Welcome to Part 4 of Chapter 11, The Functional Organization of Nervous Tissue. This presentation will cover action potential frequency and propagation, speed of conduction, and electrical and chemical synapses. Let's begin with learning about action potential frequency and propagation. Stronger stimuli result in greater frequency of action potentials. The image shows a pink subthreshold stimulus creating a small depolarization that does not meet the threshold and therefore does not create an action potential. The gray stimulus shows one action potential. The large blue stimulus produces 3 action potentials. The magnitude of all of the action potentials are the same due to the all or none principle we discussed in the previous video. The target of the action potential interprets the increased frequency as a stronger stimulus and reacts accordingly. For example, if the target was a skeletal muscle, the skeletal muscle would produce a stronger muscle contraction in response to a greater frequency of action potentials received. In order for one body part to communicate to another, action potentials must travel from the axon hillock to the axon terminals. Unlike graded potentials, action potentials do not lose their strength as they continue down the length of the axon. Action potentials travel down the axon in 2 different ways, either by continuous or by saltatory conduction. The method the action potential uses depends on whether the axon is myelinated or not. Continuous conduction occurs in unmyelinated axons, whereas myelinated axons employ saltatory conduction. Both methods of conduction travel only one direction, from the axon hillock to the axon terminals. This is due to absolute refractory periods of the previous area preventing another action potential. Also, both methods use voltage-gated sodium channels to propagate along the axon. This image compares continuous and saltatory conduction methods. On the left side is the continuous conduction along an unmyelinated axon. The right side shows saltatory conduction on a myelinated axon. Both methods begin at the axon hillock, where voltage-gated sodium channels open. Sodium enters the axon and once inside, diffuses away, creating what is known as a local current. Let's focus on the continuous conduction shown on the left. Due to the absolute refractory period, the local current only opens voltage-gated sodium channels down the axon towards the axon terminal. Those open and create its own local current to the next voltage-gated sodium channels down the axon. For unmyelinated axons, this process continues until the action potential reaches the axon terminals. Now let's look at the right side of the image where we see saltatory conduction along myelinated axons. Saltatory conduction starts the same way with voltage-gated sodium channels opening, sodium entering the axon, and a local current being created. However, the lipids of the myelin act as an insulator and force the local current from one node of RNVA to the next node of RNVA, where the voltage-gated sodium channels open. The action potential seems to jump from node to node, increasing the speed of conduction. Compare the bottom 2 images to see how far the action potentials moved in 10 milliseconds. With continuous conduction in unmyelinated axons, the action potential traveled less than in the myelinated axon. Let's now continue by learning about the speed of action potential conduction. When it comes to the speed of conducting an action potential, a few factors come into play. As we just mentioned, myelinated axons conduct action potentials faster than unmyelinated axons. The diameter of the axon also matters. Larger diameter fibers have more voltage-gated sodium channels, so they transmit action potentials more quickly than smaller diameter fibers. There are three nerve fiber types: type A, B, and C fibers. Type A fibers are the fastest, and they have the largest diameter and the most myelin. Type C fibers are the slowest and have the smallest diameter with no myelin. Type B fibers are in between A and C in their speed of transmission, diameter size, and myelination. Let's now tackle our last topic by comparing electrical and chemical synapses. A synapse is a junction between two neurons or between a neuron and an effector. Presynaptic cells is the cell that transmits the signal towards the synapse. A postsynaptic cell is the target cell receiving the signal. There are two types of synapses, electrical and chemical. The first type of synapse is the electrical synapse. Here, gap junctions connect two adjacent cells. The local current of one cell flows to the adjacent cell where sodium ions pass through the gap junction. The action potential is subsequently created in the adjacent cell. Electrical synapses are fast and synchronized groups of cells. Chemical synapses are like the neuromuscular junction we learned previously. These use chemicals called neurotransmitters to create an action potential in the postsynaptic cell. The influx of calcium into the axon terminal of the presynaptic cell causes neurotransmitters to be released from the synaptic vesicles. The neurotransmitters diffuse across the synaptic cleft and bind to receptors on the postsynaptic neuron. The binding of neurotransmitters cause the opening of ligand-gated ion channels. If the ligand-gated channels are sodium channels, a depolarization is created when sodium enters the postsynaptic neuron. If the ligand-gated channels are potassium or chloride channels, then a hyperpolarization is created. Chemical synapses allow for one-way transfer of information due to the transmitter only being able to act on the postsynaptic cell. Chemical synapses are also slower than electrical synapses because there is a synaptic delay caused by the neurotransmitter having to diffuse across the synaptic cleft. This slide lists the steps in neuron communication. Step 1, graded potential. Stimulation of a neuron initiates graded potentials in the dendrites. These are usually due to the opening of ligand-gated sodium channels. Step 2 is the depolarization at the axon hillock. When graded potentials cause the membrane potential to reach the threshold level, voltage-gated sodium channels open and sodium enters the neuron, resulting in rapid depolarization. Step 3, repolarization. Sodium channels inactivate and potassium channels open, allowing potassium to leave the cell, causing repolarization. Step 4, action potential propagation. The action potential at the axon hillock creates local currents, which bring the next area to threshold, propagating the action potential down the axon to the presynaptic terminal. Step 5, synaptic communication. Action potentials at the presynaptic terminal cause the release of a neurotransmitter, which alters the activity in the postsynaptic cell. This presentation covered action potential frequency and propagation, the speed of conduction, and electrical and chemical synapses.